Hello everyone, Gauri this side. I hope you all are doing good. In this session, we will be exploring overview of genetics and its potential scope, and it will cover history of genetics, its timeline and application. Genetics is a field of science that includes the study of inheritance and genetic variations by investigating the DNA, genes, genome, chromosome, and other components of it. Genetics research studies how individual genes or groups of genes are involved in health and disease. Understanding genetic factors and genetic disorders is important in learning more about promoting health and preventing disease. The science of genetics began in the 1800s when Gregor Mendel figured out how traits are inherited by studying peas. Mendel, the first person to lay the mathematical foundation of the science of genetics in what came to be called Mendelism and is known as the father of genetics. In 1866, Mendel published his research paper describing the law of inheritance and independent assortment. Although other scientific disciplines have also expanded in recent years, none has paralleled the explosion of information and excitement generated by the discoveries in genetics. Nobel Prizes in the categories of medicine or physiology and chemistry have been consistently awarded for work in genetics and associated fields. The first Nobel Prize awarded for such work was given to Thomas Morgan in 1933 for his research on the chromosome theory of inheritance. Morgan explained that the genes are located on the chromosome, and he also experimented on Drosophila and determined the nature of sex-linked traits. William Bateson, a British biologist who founded and named the science of genetics and whose experiments provided evidence basic to the modern understanding of heredity. J.B.S. Haldane, a British geneticist, biometrician, physiologist, and popularizer of science, who opened new paths of research in population genetics and evolution. Barbara McClintock, American scientist whose discovery in the 1940s and 50s of mobile genetic elements or jumping genes won her the Nobel Prize for Physiology or Medicine in 1983. The rapid development of genetics from Mendel's monastery garden to the Human Genome Project and beyond is summarized in the timeline. Mendel's work was rediscovered and it was correlated with chromosome behavior in meiosis in 1900s. Recombinant DNA technology was developed in 1970s and 1980s along with DNA cloning. In 1953, Watson saw that the essential DNA components, 4 organic bases, are linked in definite pairs. This discovery was the key factor that enabled Watson and Crick to formulate a molecular model for DNA, a double helix. The Human Genome Project is one of the greatest scientific feats in history. The project was a voyage of biological discovery led by an international group of researchers looking to comprehensively study the entire DNA, known as the genome, of a select set of organisms. Launched in October 1990 and completed in April 2003, the Human Genome Project's signature accomplishment, generating the first sequence of the human genome, provided fundamental information about the human blueprint, which has since accelerated the study of human biology and improved the practice of medicine. Today, genetics touches all aspects of life, bringing rapid changes in medicine, agriculture, law, biotechnology, and the pharmaceutical industry. Physicians use hundreds of genetic tests to diagnose and predict the course of disease and to detect genetic defects in utero. Scientists employ DNA-based methods to trace the path of evolution taken by many species, including our own. Farmers grow disease-resistant and drought-resistant crops and raise more transfer techniques. Law enforcement agencies apply DNA profiling methods to paternity and murder investigations. The biotechnology industry itself generates over 7 lakh jobs and $50 billion in revenue each year and doubles in size every decade. Genetic techniques are used in medicine to diagnose and treat inherited human disorders. Knowledge of a family history of conditions such as cancer or various disorders may indicate a hereditary tendency to develop these applications. Cells from embryonic tissues reveal certain genetic abnormalities, including enzyme deficiencies, that may be present in newborn babies, thus permitting early treatment. Many countries require a blood test of newborn babies to determine the presence of an enzyme necessary to convert an amino acid, phenylalanine, into similar products. Phenylketonuria, which results from lack of the enzyme, causes permanent brain damage if not treated soon after birth. Gene therapy is based on modification of defective genotypes by adding functional genes made through recombinant DNA technology. Bioinformatics is being used to mine the human genome for gene products that might be candidates for designing pharmaceutical drugs. Agriculture and animal husbandry apply genetic techniques to improve plants and animals. Breeding analysis and transgenic modification using recombinant DNA techniques are routinely used. Plant geneticists use special techniques to produce new species such as hybrid grains and plants resistant to destruction by insect and fungal pests. Thank you. I hope you enjoyed watching the video.